Okay, now we'll talk a little bit about bacteriophage, or viruses that can infect bacteria, the life cycle of a bacteriophage. And this is actually a little bit similar to HIV, as a matter of fact. So remember, viruses have to attach. So this bacteriophage that looks like the lunar lander here will attach to a host cell protein, and the host cell in this case is a bacteria. And what viruses generally use is they use receptors on host cells that the host kind of needs. So the host can't really mutate out of the virus infecting it because the host needs, for example, a glucose receptor or a sugar uptake of some sort. So this particular virus uses a receptor for probably lactose or some other sugar uptake. In this particular genome actually is kind of a neat thing. This sheath here contracts, and when it contracts, it actually injects its viral DNA into the bacteria. And then once the viral DNA is inside the bacteria, it decides, depending on the conditions of the bacterial culture or wherever the bacteria are living, it can enter a lytic cycle where the bacteriophage replicates quickly and kills the host cell. Usually it bursts out of the host cell. Or it can be kind of sneaky, and it can undergo what we call a lysogenic cycle. Okay? So in this case, the bacteriophage integrate into the genome and they become quiescent. And once they integrate into the genome, it's called being a prophage. And at a later time, it can reactivate and become lytic. So once again, the decision to become lytic or lysogenic is environmental. So if bacteria are growing and everything is good, everything is happy, it's actually going to kill the bacteria because then it can replicate lots more of itself in live, happy, viable bacteria that are growing. But if conditions aren't so good, it may want to slip itself into the genome and say, well, I'm just going to wait to see what happens here and see if conditions are good again. And then if the bacteria replicates slowly perhaps, or just kind of rides along with that bacteria until conditions get good, then it reactivates itself and becomes lytic. So this is a picture of what happens. So the phage binds to a receptor on the virus, injects its genome into the virus, and if it enters the lytic cycle here, it starts making more of its capsid proteins, its sheath proteins, tail fibers, and then is released from it lyses the bacteria. But if it's lysogenic, it actually integrates into the bacteria. And when it integrates into the bacteria may divide very slowly, conditions may not be so good, or the bacteria just may be kind of stuck. And then when conditions get good again, it can pop itself out and undergo the lytic cycle again, releasing more phage into the environment, able to infect new bacteria. Okay, so there's a video for this that you're welcome to watch. Here is the website here. And I think it goes over this on the video, so I won't spend time with this. Here's a little review here. So animal virus life cycles. Viruses make proteins with host cell ribosomes. So on an exam, if I ask you anything about a viral ribosome, you're going to immediately know that a viral ribosome there's no such thing as a viral ribosome. Viruses use host cell translation machinery to make their proteins, and that's kind of what the virus does, right? It subverts the host cell machinery. Translation occurs in the cytoplasm just like it does with any other host cell protein. It goes through the endoplasmic reticulum, the Golgi, just like any other host cell protein. Some of the nomenclature that we use in virology is the first genes, not terribly original. The first genes that are transcribed and translated are called early genes. We call them, like the first one we find usually in a virus, we call it E1 for early 1. Second one we find, early 2, not terribly original. And then the genes that are transcribed and translated later in the viral life cycle we call late genes. And typically early genes are polymerase genes because the virus wants to replicate its genome, and structural proteins like capsid proteins, envelope proteins, typically are called late genes. So you already know when a virion assembles, the genome inserts into the capsid, and this will be either in the cytoplasm of the nucleus, depending on whether it's a virus that replicates in the nucleus, like papillomavirus, or whether it replicates in the cytoplasm, like poliovirus or hepatitis A virus. And then envelope proteins are kind of inserted into the membrane, but remember that membrane is either the nuclear membrane or the cell membrane. Okay, so we already kind of talked about lysis of cell and budding. So generally, viruses that don't have an envelope lyse the cell, so they just burst the cell. Pox virus does that, it just bursts the cell. Viruses that have a membrane generally bud through the membrane. Okay? So, viruses are really, really small. We can't see, bacteria are small enough. E. coli, you need oil immersion for E. coli. Viruses are, 100 times to even 1,000 times smaller than bacteria. And it's hard to see them even with an electron microscope. So how do we figure out how many viruses there are? Because sometimes we need to, what we call titer viruses. So remember, titer is concentration. We can use also titer as a verb. We need to titer a virus. So we need to figure out how many viral particles there are. So animal viruses and bacteria viruses can be cultured within. Okay, so animal virus can be cultured within whole animals by serial inoculation, but generally we don't do that too much anymore because animals, it's very expensive to keep animals, but we can grow viruses in cell culture. And when we grow viruses in cell culture, we can figure out how many viruses there are. Some viruses actually don't grow in cell culture, and then we have to use whole animals, and that's very, very expensive to figure out how many viruses we have if we have to use, to quantitate the viruses if we have to use whole animals. Anyway, so this is what a monolayer of cells would look like if we grow it on plastic. Some of you may have seen this if you're working in laboratories. And then if we add virus to this, a virus that will infect these cells, then what happens is the virus starts doing weird things to the cell. So at 5.5 hours, some of these cells that are infected are rounding up. They're not these leaf elongated cells anymore. And then at 8 hours, the cells are kind of detaching. So the virus is completely taken over the cell. The cell is detaching. Then at 24 hours, the cells are just kind of lysing, and there's a lot of free virus here. So what's happening? This is what's happening. So we have our monolayer of cells, and we add virus. And if a virus gets inside of a cell, it's going to replicate. And typically what we do is we add some agar on top of this so the virus doesn't go up and spread outward to other cells. The virus actually spreads to its neighboring cells. And this is called a plaque, where the virus has spread to neighboring cells. And these clear areas here are called viral plaques. And we can count the plaques, and we can say that one plaque was caused by one virus. Okay, so this blue here means that the cell, we have a special stain for cells. Different types of stains for cells. Most of them are blue. So the cells themselves stain blue. And then when we add virus kills the cells, and then we don't have any blue staining anymore. Okay? So the way it actually works is we add the virus infects the cells, then we add some agar that doesn't allow the virus to swim around. So the virus infects neighboring cells, and then we stain all the viable cells. So it's a viable cell dye. We stain all those viable cells blue, and if there are no viable cells, then we have plaques here. So in this case, there's lots of virus here. There's fewer virus here. This is probably a tenfold dilution. This is a tenfold dilution of this probably, and then another tenfold dilution here where we don't have much virus at all. Here's a case where we have lots and lots and lots of virus, less and then less. So that's the dilution. So this is how we make dilutions and how we count viruses. We usually make tenfold dilution. So if we have something from a patient, for example, let's say this is from an Ebola patient and we think they have lots of virus, in a facility where you're in one of these bunny suits that's connected with oxygen, if you're working with Ebola, because it's a very dangerous virus, you would make dilutions of the virus into a medium, usually a buffer or a cell culture media. So the first one, you start out with your pure virus stock or your serum, something like that, and you make a tenfold dilution, okay, so 10 to the minus 1. And then you make another tenfold dilution, 10 to the minus 2, so that's a 100-fold dilution. 10 to the minus 3, that's a 1,000-fold dilution. You keep making dilutions, and towards the end of the dilution, then you can say, well, I think I've probably made enough dilutions, and sometimes you have to do these experiments. So this is 10 to the 5th, you plate a tenth of a mil, so 100 microliters, and you get maybe too many to count. You can't count all these. So luckily, you've also plated a millionfold, a tenth of a microliter, or a tenth of a mil, here, 100 microliters, and now you can count what, about 15 plaques there. So you say, okay, well, I have 15 plaques diluted 1,000,000 times, but I only used a tenth of a mil, so that means if I used a whole mil and got 15 plaques, that would be 15 times 10 to the 6th. So I would have 1.5 times 10 to the 7th viruses, but in this case, I only used a tenth of a mil, so I have 1.5 times 10 to the 8th, or 15 times 10 to the 7th, or 1.5 times 10 to the 8th. You can move the decimal back and forth however you like. Okay, so last clicker question in the last slide here. RNA viruses utilize blank for replication. Go ahead and stop the video and figure out what the answer is. And the answer is a viral RNA-dependent RNA polymerase. And the reason why this is correct is because this is an RNA virus. Okay, so since it's an RNA virus, it has to make its own RNA-dependent RNA polymerase because we are not made of RNA, we're made of DNA, okay? If the question here was DNA viruses utilized, then you might say a host, well, the answer is, so E would be a host DNA-dependent DNA polymerase for replication. So the answer is not even up here if it was a DNA virus. Okay, so that's it. Make sure you watch the next lecture as well, which is Virology 2, and I'll see you in class.